Welcome to the THOR Table of Knowledge podcast, where we dive deep into the groundbreaking world of trauma care, hemostasis, and oxygenation research. A special thank you to our sponsor, Velico, for their support. Velico is revolutionizing trauma care with their innovative frontline ODP spray-dried plasma system, with the goal of empowering blood centers worldwide to produce dried plasma for transfusions.
Good afternoon, THOR listeners. My name is Andre Kapp. I am a member of the THOR Executive Committee, and I'm speaking to you today from the Solstrand Resort in Norway, where we are finishing the THOR 2025 annual meeting. It's been a great meeting. Today I have the distinct honor to share with you some groundbreaking research and introduce you to my friend and colleague, Dr. Anirban Gupta, Professor of Biomedical Engineering at Case Western Reserve University. Anirban, your work most recently has focused on the problem of platelets. We don't have enough platelets in the world, right? Not enough platelet donors. They're hard to store. They're super important for hemostasis. Stopping bleeding is the number one goal, really, in the care of trauma patients who are bleeding. And so the most critical blood product is in the shortest supply. And sometimes they don't even work that well because of the way they're stored. So you've hit this problem head on and said, well, maybe we can engineer something to replace or at least augment platelet function. So tell us a little bit about your work. What's the project?
Sure. And thank you. It's a privilege and honor and I have known Dr. Kapp for a long time now. I stopped counting because it's a privilege to know you as a friend and a collaborator and learn from you. So I'm a materials engineer by training, not trained in the blood field, but found sort of my calling in the blood space by speaking with folks like you. I started in the blood space. The research was about preventing clotting, not promoting clotting. And I was building coatings that mimic the natural way that body prevents clotting on cells in a healthy environment. And these coatings were directed at designing ECMOs and other sort of blood contacting devices where clotting is a problem. But that prompted the thought that if I could engineer materials to prevent clotting, what about the other end of that spectrum? Mm-hmm. Can we design materials to promote clotting? When I had started thinking about this, trauma was not the focus. It was more about thrombocytopenia, other platelet defects. So it was a very platelet-centric problem to begin with. I think that in retrospect created a lot of these design refinements and modifications as we thought about our research. Very serendipitously, during these thought processes and early development, I got to meet, including you, a lot of folks, Dr. Phil Spinella, Dr. Jason Sperry, Dr. Mackie Neal, to name a few ranger medics that I don't know the names of at different defense meetings, that all in different ways underscored the context of bleeding control in trauma care. And that really opened my field of view, so to say, where the potential of these things can go. So then we looked at, artificial platelet field, and we realized that the terminology of platelet substitute is a pretty old terminology. There have been publications going back to mid-1990s using that terminology. But all the designs essentially focused on what I call a superfibrinogen molecule. So they were only looking at platelet aggregation, and then they were taking particles and coating them with fibrinogen or fibrinogen fragments to create an enriched platelet cement, is how I think of it.
I think that's fair.
Which is scientifically pretty cool for the time that it was happening. But platelets, as I always say, are like drones. They surveil, they home in, then they do their thing. So if we go by that model, you have to design in this homing signal for it to then do the rest of the functions that they do. So it's a multifunctional drone, and that's the the design model or design motivation with which we looked at a synthetic platform and then modularly mimic how platelets surveil and attach, then how they co-aggregate with each other, and then how they augment coagulation in a very site-specific way, and then how they stabilize clot, again, in a very site-specific way, so that we can avoid off-target harmful side effects while augmenting targeted hemostatic effects. So that's sort of the 50,000-foot view with which we started our research program more than a decade ago now. But
It's been a minute,
right? Yeah, but it has been.
I remember.
Yeah, it has been a really great learning experience, and every time I come to THOR and other like ISTH and other meetings that I go to speak with the clinical side of where such treatments could become useful, but also the basic science side. And then as new discoveries happen in the platelet and coag field, we try to leverage that knowledge into refining our design as needed to really make a customizable artificial platelet platform.
Well, you're taking the synthetic platelet concept to a level of sophistication that obviously nobody's ever done before. So I think the odds of this yielding a useful product are much higher than ever before, really. Part of your,
I hope so.
I mean, I hope so too, because God knows we need it.
Yeah.
Right. This is be a big deal. So you received some funding from DARPA as part of the F-Sharp project, which is a really fascinating attempt by the agency that likes to sort of fund high risk, high reward, groundbreaking projects, right? Talk to us a little bit about F-Sharp and what the components are and the goals, and we'll pursue that a little bit.
Sure, absolutely. So I would like to backtrack a bit in order to get to the DARPA thing, and that kind of connects with THOR. So again, when we started doing the plan, you remembered this because you have seen many of our studies, both in publications and posters, and we have collaborated. We started our research not envisioning a powdered product. We started our research envisioning a liquid sterile solution because, again, pre-hospital was not at the foreground of our thought process. And then I was fortunate enough to come to THOR and also MHSRS, where we have met up many times, and speak with you and others, where we were asked questions like, oh, can it be a powder? Can you take it outside? How long does it stay stable? Can you put it in an auto-injector? Can you give it intraosseously? All of these sort of ConOps, logistics, field use questions that essentially prompted us to investigate many of these answers with DoD funding. So the original fundings came from MRDC in the hemorrhage control space to get it to the point where we were successful in developing a freeze-dried version of our artificial platelets that reconstitute under a minute. It's stable at room temperature and 4 Celsius for at least a year. It's stable at 50 Celsius for at least 2 months. And you can design an auto-injector around it and make it a combined device product. So there were a lot of logistics things that sort of fell in place around the time when the DARPA endeavor came around. And the DARPA endeavor came around actually during COVID, which was a great relief because I was getting lazy because we were not doing research. Things were kind of shut down. So when Dr. Commander Kratian connected with me and then with Phil and you and Mackie and many others, there was a lot of discussion regarding artificial blood because there has been a pretty, I would say, nuanced history of biosynthetic blood products in the hemoglobin-based oxygen carrier space.
Exactly.
And the perfluorocarbon space. So while those products were developed with the thought process of artificial blood, they were not artificial blood, they were artificial oxygen carriers. And they had their own pluses and minuses. Ooh. We all kind of know about it and we don't need to get into it here. But the new thought process became that, all right, if somebody is freeze drying, or now in Velico's case, spray drying plasma, if somebody is making a powdered version of a platelet mimicking technology, and if somebody else is making a powdered version of a hemoglobin-based oxygen carriers, is there an opportunity to integrate them together and
Right.
make a biosynthetic blood powder, which has a small form factor, small carry weight, can be very portable, hopefully stable for required amount of time, and then the thing becomes just add water as needed. So it's an on-demand use that can be a bridge between definitive care with biologic blood products versus nothing available way far forward in the austere environment or in expeditionary medicine or in blood deserts that
Right.
you're very, very well involved in. So that's sort of the vision that emerged out of the discussion with DARPA towards the end of 2020, early 2021, which then led to the F-sharp program solicitation, and we were very fortunate to be picked up as the platelet component of the endeavor. So we are currently in year 3 of that endeavor with a goal to get to an IND by the end of year 4.
Now, so far, the company that spun out of your work at Case, Hyma Therapeutics, right, has partnered with Calicite,
Yeah.
which is making the erythromer nanoparticle hemoglobin-based oxygen carrier, basically.
Right.
And then there's a plasma backbone to this. And you've done some animal studies. Are things looking good?
Yeah, so I don't do the animal studies. They're at Pittsburgh and at Maryland. The animal studies currently are safety studies and not the efficacy, so they have not gone under uncontrolled hemorrhage and shock yet. The safety signals look good,
Great.
right? And again, I can only theorize here. We already know, so we have done safety studies with just our standalone artificial platelet product across a variety of species, including some collaboration with your lab. What we have found is that the lyophilized system has a therapeutic, so it is effective at doses as low as 0.1 mg per kg in certain animal species. And then its toxic signal is at least 250x from there. So there's a pretty good safety window for the synthetic platelet part of it. The erythromer part they are studying now in animals. The plasma part is not in our domain, so it's from a company, or it might be from multiple companies. So they would have their own safety profile analysis and things like that. The question then becomes, when you put that together, do their individual safety profiles stay preserved, or something new happens that we hadn't anticipated before? So far, so they have been put into rabbits. So far, the safety signal is very positive. So the efficacy models start actually this year, both in Pittsburgh and Maryland. And then we will see. I can't preempt, but hopefully it works.
Well, it's very exciting. I mean, there's still, I guess, a lot of work to be done on dose finding and defining that therapeutic index for each of the components separately and together.
Yeah.
And coming up with kind of that optimal combination.
Yeah.
But what we could be on the cusp of here, at least with Hyma's product, is a major breakthrough of moving to a platelet substitute or enhancer that could reduce the need for platelet transfusion, which would be huge.
Or a bridge when platelets are not available or the patient is refractory to platelets for some reason.
Oh, that happens all the time, right?
That happens all
Platelet
the time,
refractory,
right?
that's right, make antibodies.
And the other interesting thing that we have done, this is, out of the DARPA endeavor, we have made, from a research standpoint, we have made in-house, for example, FFP, following published protocols, and we have mixed in our artificial platelets in the FFP to see if it acts as a force multiplier in a hemodilution model. And it indeed does. So when you give just the particles, you get a certain positive trajectory signal. When you give just the plasma, you again get a positive trajectory signal. But when you combine them, it's an enhancement, like a booster effect, where the signal gets amplified. So this kind of creates the opportunity to think of combinatorial. So I don't want to use the term component therapy because it's still integrated.
Yep.
But it's a combinatorial therapy where you get a cumulative amplification of your outcome, which is better than each of those. But while we do these studies, we also look at their individual effects on endothelial cells, other blood cells, etc., so that the safety signal is never at risk while these cumulative effects kind of get amplified. So that creates new research opportunities and new kind of, new even product ideas, maybe collaboration or joint ventures between companies and things like that.
So just for the audience, we've mentioned these particles and what they look like. So, a platelet is a subcellular anucleate cell fragment, essentially, basically 1 to 3 microns in size, decorated with many different receptors, but to bind things like fibrinogen, as you mentioned earlier, von Willebrand's factor, and they can also interact with leukocytes and endothelial cells and so forth. Can you just give us a quick sketch of what this the platelet mimic nanoparticle looks like? How big is it? What's it got on it? How does it compare to a regular platelet?
Sure. It's actually about 100 times smaller than a regular platelet, but it has a higher surface to volume ratio than the regular platelet because it's smaller, but it's a spherical system, right? So if you do the ratio of surface area versus volume, it has a lot more surface area than a regular platelet to play with, so to say. Now, as you were saying, that real platelets do a variety of things. So again, if I go back to that multifunctional drone model, some of those things are surface mediated, some of those things are cargo mediated, right? Yeah. So that creates this sort of, different levels of multifunction. It's not just multifunction in a linear way, but multifunction in an exponential way, right? And what that means is one can modularly pick what do you want to mimic, how much you want to get that extent of mimicry, and then can you combine different mimicking functions to get a cumulatively higher effect. So if I look at sort of the simplest mechanistic way that platelets form a clot, they have a homing signal to bind to where the injury is, and then they join hands with each other like sandbags piling up to stop a flood, right? And so that's their primary hemostatic function. That's how they primarily start mitigating bleeding. So we mimicked using fully synthetic peptides on a fully synthetic lipid nanoparticle, this sticking to an injury site function and then co-aggregating with themselves as well as co-aggregating with whatever native platelets activated they find around themselves to start forming the plug. Now, if I go by that same visual of sandbags piling up, under the force of the incoming flood water, the bags can fall over. So our natural biology has created this really elegant system that the bags can make their own mortar to stabilize themselves. So platelets help generate an enzyme called thrombin, which generates a protein fibrin from another blood protein, fibrinogen. So fibrin is the mortar, or in case of a sandbag, the nylon netting that integrates with the bag to stabilize them. So while our generation 1 artificial platelet is a simple lipid nanoparticle decorated on the surface with injury site binding and then injury site aggregating capability, our generation 2 takes that platform and then incorporates a function that allows generation of thrombin, but again in a very site-specific way, so that now the generation 2 is capable of making its own mortar. So it's making fibrin where needed and not willy-nilly in circulation. And then generation 3 is essentially mimicking platelets' nuanced granule function. Now I consciously use the word nuance because depending upon who your patient is, you need certain granules contents to be secreted. You don't want certain granule contents to be secreted because there can be inflammatory side effects. In our synthetic system, you can pack what you need.
Sure.
Right? So you are in control. So the whole point of a drone is if you can't control a drone, it's useless. Right? So that's what we try to do, is to pack. So we have packed tranexamic acid inside it. We have actually packed thrombin directly inside these particles and bypassed the whole coag cascade. We are now doing studies with packing inorganic polyphosphates, which is a platelet granule content, but you cannot give it directly because it has its own inflammatory risks. But if you package it inside a particle such that it only gets released where the particle goes and binds, now you're controlling where you're releasing the cargo and you're amplifying the effect. So it's like starting from a simple lipid nanoparticle, not very different from the vaccine particle or any of the other sort of doxil is the earliest lipid nanoparticle that was approved in 1992 or something for chemotherapy. So the actual lipid composition is not very different from all of these already approved particles. The unique aspect is the peptide decoration and the engineered lipid incorporation for procoagulant function, or the cargo that may get incorporated as a packaging entity inside the particles.
So how do you get it to release the cargo?
Great question. So now we are actually getting into back to my materials engineering degree. So we work all the time on enzyme responsive materials, not just with artificial platelet, but in a variety of other materials engineering things that we do. And the fundamental idea is that there are certain enzymes that either are secreted or upregulated in a site-specific way, and as they dissipate from the site, they get inhibited, so they get neutralized. So away from the site.
So thrombin, for example.
Thrombin, for example. It's a perfect example, right? So away from the site, their activity is low. At the site, their activity is high. So if you take thrombin or plasmin from TPA getting produced from injured endothelial cells as an example, we can use these as triggers to potentiate a function, right? And so what we have done is we have created very unique lipid systems in the shell of the particle that actually degrade under the action of things like thrombin or like plasmin. So the particle with the cargo is intact in circulation, and then they come and bind, and that spike pokes holes in the particle, and the cargo gets released out.
Brilliant. Well, Anirban, this has been absolutely fascinating. I hope our listeners have gotten a really good peek into the future.
I gotta give big shout out to my team, right? Like, I get the privilege to talk to you about these ideas and products and companies and whatnot, but it takes a village, right? And so, both on the company side and the university side, collaborators and researchers and clinical advisors, it's a very diverse team of expertise that makes all of these happen. Some people help us identify the problem. Some people help us, come to a design prototype. Some people help us execute the design prototype. So it's really a closed loop system that makes all of this happen.
It's super exciting work. I feel incredibly lucky to have had a teeny little role in collaboration with you over the years. But the creativity is amazing. So listeners, we're not there yet. These projects take a long time, but we are making progress towards, I think, really a breakthrough technology here. So Anirban, again, thank you so much.
Thank you so much for the opportunity. And I'm reachable anytime.
There you go.
And the generation 1 artificial platelet is actually in a fast track to get into safety clinical trials by next year. So
fantastic news.
Yeah, while things do take a long time, we have been already doing a lot of the backdrop work so that we are now very well positioned to get into our phase 0, phase 1 trials by next fall. And then, if that trajectory continues, these things could get to a patient in a couple of years. So
fantastic.
We'll see what happens. So.
Well, innovation brought to you by THOR. So thanks very much, and we'll pick it up as you make progress on the project.
Thank you so much. I appreciate the opportunity and happy to chat anytime.
That's a wrap for this episode of the THOR Table of Knowledge podcast. A big thank you to our sponsor, Velico, for supporting the advancement of trauma care. Their frontline ODP spray-dried plasma system is paving the way for blood centers globally to produce potentially life-saving plasma for transfusions. Check out our website, www.thornetwork.com, for more podcasts, and join us next time as we continue to explore the cutting-edge science of trauma, hemostasis, and oxygenation. Until then, stay safe and stay informed.